Hello everyone and welcome to our poster, Untangling the Graph Structure of Drosophila Brain Datasets with Open Source FlyBrainLab Utility Libraries. I am Mehmet Kerem Türkcan, a PhD candidate at Columbia University in the Department of Electrical Engineering, and this is joint work with my advisor Aurel Lazar and Ian Zhao. In this presentation, I will be highlighting our open source utility libraries. You can see two links here. FlyBrainLab installation instructions can be accessed via the first link, and the utility libraries can be accessed via the second. For the fruit fly Drosophila, recent releases of large-scale connectomics and synaptomics datasets have started to enable us to explore neural circuits in unprecedented detail. We have created the FlyBrainLab open source interactive computing platform for studying the function of executable circuits constructed from such data. FlyBrainLab, within a single working environment, provides 3D exploration and visualization capabilities for FlyBrain data and a means for creation of executable circuit diagrams from the explored data, as well as an interactive exploration of the functional logic of these executable circuits. FlyBrainLab user interface enables us to explore the connectivity of neurons and synapses, visualize the circuit diagrams that are automatically generated or drawn by hand, and use a notebook for program or subroutine execution. The complexity of the connectomics data, even as graphs, can be intimidating. Our utility libraries help untangle the graph structure of these neural circuits. Here I will highlight NeuroEmbed, NeuroSynapses, and the NeuroGraph libraries. The NeuroGraph library offers a set of tools to discover and analyze any connectivity pattern among cell groups within a circuit. We allow for the generation of dendrograms for any queried subcircuit of interest. We have also integrated popular community detection algorithm libraries in Python for use with FlyBrainLab circuits. We have also integrated methods to discover driver nodes and compare existing graphs against random graphs. Here, we show the Louvain algorithm applied to approximately 20,000 neurons in the hemibrain dataset. We observe a number of communities that correspond to known structures in the brain, and on the right I have visualized neurons according to the clusters that they were grouped into. Secondly, we have the NeuroEmbed library, which implements a set of algorithms for structure discovery based on connectome graph embeddings into low-dimensional spaces. With NeuroEmbed, users can also use morphometric features such as axon length and thickness, width, volume, and surface area to group neurons in a subcircuit of interest. Based on the embeddings, users can find similar neurons given a neuron. Most importantly, with the NeuroEmbed library, we have sought to provide a standardized benchmark for many different tasks that embeddings could be used in so that database bias and comparison of such methods could be alleviated. Here we show a two-dimensional embedding generated via NeuroEmbed for the VA1V glomerulus dataset, which shows that it was able to correctly separate the different cell types. For new datasets or for trying to find similar neurons in a subcircuit of interest, NeuroEmbed can generate such embeddings with only a line of code. This embedding greatly simplifies the understanding of the circuit, for which the 3D visualization of the morphology is shown to the right and is hard to make sense of. To the right in Table 1, I show the different embedding approaches for different tasks. We compare 4 embedding algorithms in various tasks: similar neuron search, discovery of cell types from connectivity, and generating a labeling of cell types given incomplete labels. For the discovery of cell types, we use adjusted rand index, that is ARI, and adjusted mutual information, that is AMI matrix. We employ twofold stratified cross-validation accuracy, ACC, for semi-supervised classification. We choose to use the classical major cell type studied for the relevant datasets for these tasks. As you can see, we see different approaches as being good for different tasks on different datasets. Lastly, we compare the usefulness of adding anatomical morphometric features on top of graph embedding-based features in Table 2, and we observe that the combination of both helps semi-supervised learning accuracy. Finally, we have the NeuroSynapses library, which offers a large set of utilities to accelerate the construction of circuits and analysis of connectomic and synaptomics data. NeuroSynapses helps users retrieve neurons and synapses even through multi-hub connections. It also allows for statistical analysis of specific synapses. Here, I have 2 examples from the NeuroSynapses library. On the left, I show a clustering of the synapses in the mushroom body with K-means cluster centers highlighted. Those who know the shape of the mushroom body neuropil can immediately see it here. These algorithms can be applied at neuron partner scale or whole neuropil scale, as I'm showing here, and results can be obtained in short amounts of time. say under a minute. On the right, I show a simple one-liner function from the library that calculates synapse densities on a per neuropil level for synapses that we have pruned based on a characteristic we were interested in, say after filtering out connections with low numbers of synapses. Thank you for watching, and I'd love to answer your questions and tell you more about our work. Thank you.